Okay, it's great to be back. So I'm going to tell you about other work in the lab. And before I forget, I just want to say there's two posters on this, and I, of course, was not as organized as John. So on the first part of the work, a poster is going to be presented by Tammy Reed, and that'll be up later today. And then the second little story I'll tell you about, Subodh Mishra, an MDF fellow, will be presenting that work in a poster as well. So if you have questions, they're great people to chat to about the projects, and I'll jump forward. So just in terms of thinking about therapeutic strategies, there's lots of sort of different areas. This is a review by one of my colleagues, Kailak Reddy here, who's here as well, and you can ask him questions about some of the approaches, and I'm going to present some of his work as well. So we think about, the disease mechanisms of myotonic dystrophy. There's the different bullets tell you places where you can sort of target. And I'm going to tell you mostly that we try and target as far upstream as we can. So we think a lot about targeting transcription, things that would reduce the toxic RNA. So those are the two stories I'm going to tell you primarily about. We do sometimes seem to bump into the upregulation of MBNL, and that's part of story one as well. So we've been actually at this for quite a while. Back when I was at the University of Oregon, a couple of different students worked on screening small molecules. We primarily focused on FDA-approved molecules as well, and we hit upon pentamidine, an anti-malaria compound that we published in collaboration with Charles Thornton, and we demonstrated that this small molecule could rescue splicing, but it was at high concentrations, and the therapeutic window was very small. We were rescuing, but probably also basically killing the mice while we rescued. So not an optimal compound, but as a proof of concept, it was good. So we kept moving down this pipeline, and we kept screening what we call diamidines. We hit on heptamidine, and we published that in 2013. Heptamidine, we improved the rescue of splicing. We still had some toxicity, but our window of therapeutic window had opened up a little further. And then as we continued to screen in this class of diamidines, we hit furamidine. And this is something we published in 2018. We have a couple other publications, and this is by an MDF fellow, Jenna Janklin. And so she showed that furamidine could rescue missplicing, and really we weren't seeing much toxicity at all there. And this is another anti-malaria compound that had made it to phase 3 clinical trials. It does still have some safety issues, but we felt like we were sort of moving in the right direction. And then just recently, Jana published in 2020, another diamidine a little larger, and this compound 7, it doesn't have a fancy name, rescues a little better. So this all sort of has prompted us and motivated us to keep looking in this class of compounds. And Hormoz, Hormoz, and I won't say his last name because I always butcher it, a medicinal chemist who's part of the group, really sort of dove into the data and thought about the molecules, and he asked some important questions about how we might make additional new modifications. And what Hormoz has done is he's modified using the furan core, this heterocyclic core, to make new molecules, things that haven't been made yet. And we've tested those. And so on Jesus's poster, and Tammy will present that, you can see more of the data. I'm just going to hit some of the highlights. And tell you that we've identified and I forgot to change this, of course, that highlighted in yellow is not supposed to be there. So when we call these compounds modified polycyclic compounds, so you have to come up with an acronym, and this is due to John Cleary, who's here, so you can talk to John as well. And what we were really excited about is what you can see here is most of our compounds were in the micromolar, maybe low nanomolar, and now with Hormoz's modifications, we've gotten into the low nanomolar. So those are sort of the concentrations that, at least when you talk to drug companies, they get a little more excited about your compounds. So these, for Hormoz's initials, Hormoz has made a series of compounds, and Jesus tests them in cell models. We start in DM1 patient-derived fibroblasts because they're easy to work with. And you can see in the dose curves that you see, splicing rescue in the low nanomolar. And actually, as you go further out, it sort of comes back down, probably because of a lot of off-target effects. But we have nice dose response curves. And I should say what I meant to put in here was that there's no cell tox until the micromolar. And actually, I just didn't include the data due to time. We also use patient-derived myotubes. the muscle cells. And here, higher concentrations of the compounds are required. It's not always the case. It depends on the mechanism of action. In this case, based on our mechanism, which we initially thought we were targeting the toxic RNA, and we think that's an aspect of this, but it's something we're still teasing out. So these compounds, now we're in the higher nanomolar concentrations. It's probably because there's higher toxic RNA concentrations that we're targeting in the myotubes. And again, you see a nice dose response curve for two different splicing factors and two different compounds. HM33 is a compound that we're excited about because based on the modifications is predicted to cross the blood-brain barrier. So the goal here is to make molecules that can be delivered systemically and cross the blood-brain barrier. And we think, just like with furamidine, the other some of these diamidines, and I should say these aren't diamidines, these are sort of a new class of compounds. They we do see some reduction of the toxic RNA, but we also see this interesting upregulation of MBNL1 and MBNL2 transcripts and concurrently with the proteins. So we're excited about this class of compounds. We have a lead MPC that rescues splicing. I don't have to introduce the ATP2A1 channel because John did a nice job with that. And at daily dosing for 5 days, we see dose-dependent rescue of the ATP2A1 exon. We need to continue to explore formulation delivery of this MPC. It's not sort of a perfect molecule that just goes right in. So we have work to do there, and we are working on PK and PD studies. We've just done RT-PCR on a few of the splicing markers. We needed to perform RNA-seq and do the functional assays, but we're still sort of early days on these MPCs. So that's the first story, and that's an exciting part of the lab. I want to tell you about a second part, and again, I didn't update my slides. So we've synthesized novel small molecules, as I said. We think there's some crossover for DM2. I didn't have time to tell you about that. I'm not sure it's on the poster, but you can ask Tammy or others. And so we're continuing to test new compounds in DM1 cell lines to do structure-activity relationship studies. And we're continuing to explore the mechanism of action and best routes of delivery and formulation. I'm going to tell you about a second story that was in collaboration with Eric Wang's lab at UF. And this is work Ona McConnell developed this cell line. It's been sort of a workhorse cell line that we've used multiple times. So I'll just say that it's a really fun cell line because what you can do is you can look for compounds or factors that selectively reduce your toxic RNA. So using Tom Cooper's model, this was integrated. We tend to use the 480 because the 960, we have a little bit of issues with expression levels, but the 480 gives very robust, reproducible expression levels. And then we have two unique sequence tags. And using those two unique sequence tags, we can look for things that reduce the long repeat RNA and the no repeat RNA. And we're always looking for things that do a selective reduction. And so just to highlight some work from Kalak and Tammy and Sabode. We've done lots of screening. It's a relatively simple screen. It's in HeLa cells, so they're easy to grow, easy to use. We treat the cells overnight at 1 micromolar, lots of different libraries. And then we use the multiplex RT-qPCR to look for compounds or factors that will change the ratio of the toxic RNA. So we're always looking for things that reduce the toxic RNA and don't affect the control. So this is published, so I'll just go through this quickly. This was work from Kalak that was published in PNAS. And what we did was we screened the low-pack library that has FDA-approved drugs. And I'm just highlighting, this is one of the repurposing compounds that Charles talked about, is we identified colchicine, something that we didn't expect to identify because we weren't thinking that microtubule inhibitors would have any impact on toxic RNA levels. But it's clear to us, and it works in cell models and in the mouse models, and that colchicine reduces the toxic RNA. So that we got it out of the HeLa screen. It translated to the cell models. It translates to the human skeletal long repeat actin mouse. And what you can see is we're seeing this reduction of the toxic RNA, a partial rescue of the splicing from the RNA-seq data, and not massive off-target effects, as you can see in panel C. And it's not affecting the mouse DMPK transcript, which doesn't have the repeats. So this was the sort of proof of principle that this approach could work. We could screen in this sort of artificial HeLa cell line. And so Sabode Mishra, an MDF postdoctoral fellow, wanted to screen natural products. He was excited about the possibility of there being natural products out there that could reduce the toxic RNA. So he took the same screen. In this case, it's from the National Cancer Institute, screened the 390 compounds from this library. And you can see the little line where there's 6 hits. They're secondary metabolites from plants and bacteria. And they reduce the toxic RNA by 50%. That's the cutoff we use. And there is some toxicity for 5 of the 6. Natural product 1, it's a modest reduction in terms of cell viability. So that was the one we decided to pursue. So what Sabot did was he did a secondary screen. So he was able to go through the literature and find natural structural analogs of this natural product 1. And so there's a whole series of them. And so Sabot went through and then redid the screen. And as you can see, there are a class of these red ones that work a little better than the other classes. And he picked this natural structural analog 1 because there's even less toxicity, and pursued that to translate that into the patient-derived cells. And so this analog, it rescues the missplicing in a dose-dependent manner of the insulin receptor, rescues the splicing of this other biomarker we use in the fibroblasts. And then excitingly, we see this nice dose-dependent reduction of the DMPK transcript. And then there's no obvious toxicity in unaffected cells or the DM1 cell line that we're using. We also want to check to see if we see the same thing in myotubes. And just like we see with some of our other compounds, because of the higher toxic RNA levels, it tends to not see the full rescue. But here we're seeing the same effect, the same dose response curve. As you get out to really high concentrations, 128 micromolar, you're starting to lose the rescue. And you can see there's sort of that sweet spot where at 32 micromolar, we're seeing a nice reduction of the toxic RNA. And it's the optimal concentration for splicing rescue. So then we took this into the HSALR mouse model. We put it just in their water. So they're just drinking the compound. It is a modification. So for stability purposes, this analog has been modified. It's in the drinking water. I should say this was done with Masayuki Nakamura, one of our longtime collaborators. And Masayuki was excited to report out the data. So here's that chloride channel in the mouse model where you can see you want to get the splicing back up to the wild type. We're not quite there, but you can see with the low dose, there's really no effect. And with the 6-week high dose and the 12-week high dose, you're seeing the splicing rescue, that ATP2A1 biomarker, splicing biomarker, you're seeing the same effect. And you can see that at the 12 weeks, we see a really nice reduction of the toxic RNA, and that's the HSA transgene that has the CUG repeats. And then a control transcript, there's really no change. So Sawyer and Sabot did RNA-seq on the muscle samples from the mouse, and you can see, you know, the HSALR long repeat is in the middle panel, and then when you've done the 12-week treatment, you see this nice shift back towards wild type. I don't think you can quite see it up there. I'm sorry, but there is a rescue of the histopathology, and then importantly, there is a rescue of the myotonia. It's not a full rescue of the myotonia, probably because we're not getting a full rescue of the splicing. And then our RNA-seq data, we're still in the process of analyzing this, because what we're doing here is we're comparing the HSALR to the 12-week treatment. And what we think is some of these changes actually are transcriptomic changes that are going back to wild type. So it's not all off-target effects, but we're still sort of parsing that out. So this is stuff we're really excited about as well. So how does this molecule work? So we've been thinking about this for a while. One of the things we did with colchicine is we showed using pulse labeling experiments where you're labeling the nascent transcripts. And so if it's working at the level of transcription, you'll see a reduction of the RNA right away. And so at a 30-minute time course, a 1-hour time course, in 3 hours, you can see with colchicine and the natural product, you see this reduction. We are seeing this little bump back up at 3 hours, and it's possible our natural product isn't working as well as colchicine across this. So that's something we're trying to understand what that data tells us. I should say we're continuing to add data points here because we're not quite done with this study. And then Subodh identified a nice assay because based on the structure of the compound, it was predicted that it might be an intercalator. It's not something that creates a lot of mutations like strong intercalators do. And so what Subodh did was he asked in this fluorescent intercalator displacement assay, where you have cyber safe as a molecule that intercalates in it, and then actually you displace it and you see this change in fluorescence, the natural product has a really nice displacement consistent with it intercalating with the nucleic acid. And this is a CTG-CAG repeat structure. So our working model is that it intercalates. And I'm not saying it's some perfect molecule that's binding just at the repeats. I actually think it's binding all over the transcriptome because we're at high concentrations. But I'll sort of give you the punchline is that I think it's something that, well, it's in your diet. You can eat lots of it. And I think because Pol2 has challenges getting through long repeats, you just put just enough roadblocks through this natural product that Pol2 is stalling out and you're inhibiting transcription. But if you look at other genes, many other genes, you don't see the same effect, even though almost certainly this small molecule is binding throughout the genome. So the working model, and I'm sorry, the tech transfer folks asked me to block this out because we haven't quite finished out the IP, is that this molecule intercalates probably throughout the nucleic acid, as I said, and we have sort of a model for how we might imagine making modifications. So we've been thinking about ways to do that and to understand the mechanism. So just to summarize the next steps, so we're excited about this. It shows a significant anti-DM activity in our studies across a wide range of DM systems. We're not seeing any obvious toxicity. As I said, it reduces the toxic RNA levels in the HeLa DM1, in the DM1 and DM2 fibroblasts. I actually have more time than I thought, so I could have shown the DM2 data. But if you're interested, Subodh has that on his poster. And it works in the DM1 myotubes, as I showed you. It works in the HSALR mouse model. And so actually, the way we've been thinking about this is that it's not working in the context of, say, the specific sort of repeats in the context of DMPK, but it actually works because there are repeats there. Because the HeLa cell, the DM, and the HSALR, the repeats are all there, but they're in very different genomic contexts. So that's the way we've been thinking about that. So we think it may have the ability to work across other repeat diseases. So as I said, the working model for the mechanism of action is that it binds the CTG, CCTG repeats. And as I said, many other locations, we're not making any claims that this is an incredibly specific small molecule. But we do think because, as I said, the challenge of transcribing through repeats, that it actually has a greater impact at the repeat locus than other things. And so one thing we want to ask is if we test things with longer repeats, do we see greater effects versus shorter repeats and do those types of mechanistic studies? Something we're just starting to ramp up and do is we're really interested in the idea of testing combination of small molecules. So one obvious one is looking at the first story I told you about, the MPCs with the natural products. But we're also going to ramp up and do a screen of natural products with sort of at a fixed concentration where we see a partial rescue, and then screen lots of other natural products or FDA approved drugs. Actually, and John and I need to talk. Maybe we need to try a combo with his molecule to see if there's any interesting activity. And yeah, so we're excited about this molecule. With Nick Johnson and Jeff Statland, we're thinking about how we can move forward with clinical trials. We'll see where that goes. Yeah. And so just to say, this has been a fun project that Subodh Mishra has really done a beautiful job. The second part, Jesus Frias with Hormoz Mastiansi has done just great work on project one. Sawyer has tied in and done a great job on the bioinformatics on the second project. This has been in collaboration with Kailak Reddy. So he's co-mentored Subodh Mishra on this project. And Kailak really started the idea of looking for selective reduction of the toxic RNAs. So it's fun to have Kailak at the RNA Institute continue to work on this. And again, a lot of this all started with Eric and Ona McConnell. Ona's here, so I hope she enjoys seeing how her work continues to build on in the field. Masayuki Nakamura, who trained with Charles, and I remember many years ago when Charles sort of said, "Hey, you should keep working with Masayuki." And so we've been doing that for now 10 plus years. So it's great. I meant to acknowledge Sweta Van Gavetti. She is our computational modeler. She's really started helping us think about how the small molecules interact with nucleic acids and doing modeling studies so we can think about potentially making modifications to improve the targeting. Yeah, and actually I forgot both times to talk about my disclosures. So I'll just say them. So consult for Kate Therapeutics, Entrada, Juvena, and Syros. And we've had sponsored research from BioMarin, Syros, and Agios. And so that's it. Thank you.
Really exciting, interesting stuff. I was curious if in the HSALR mice that you've been giving this NPI-NSAI, are you looking at any other whole animal effects beyond just looking at myotonia, like treadmill running, activity?
Yeah.
Any improvements there?
No, that's being written into a grant proposal. We've sort of been doing all this on the fly. So yeah, we need to do all the functional readouts. Completely agree.
Updates to come?
Yes.
Fabulous. All right. Thank you so much.
Yeah.
Yeah.
So, this compound NP1 that binds to CTG repeat, is it reversible binding?
Yeah, it's reversible binding. Yeah. And it's not incredibly tight binding either. It's micromolar, low micromolar. Yeah, just in that.
You said you looked at type 1 and type 2 fibroblasts. Is there a difference in this dose response to your compound in those two, since it's type 2 and such large?
Yeah, so it does take a little higher doses in the type 2. And I don't think for all the splicing biomarkers we look at for the type 2, do we see a full rescue. So it may be that there's a concentration effect there. Yeah. Yeah, Laura?
So following up on the last, you looked at the viability of the cell. Have you looked at the viability of the cells or MTT assays or anything else?
So with all the viability studies, we have to get to super high concentrations to see any effect on viability. We have a lot to do on the mouse work to see if there's we don't see any obvious tox effects. But with HSALR, Masayuki says they seem happy. That's his report to us so far. So we need to do more there, obviously.
So you said it binds to the DNA.
Yeah.
Do you think it binds to the RNA?
Yeah, it binds lots of places.
You think?
So yeah.
And I just a clarification. So NP1 came through a natural product screen.
Yep.
And NSA1 is an analog, but it is also itself a natural product.
Yes.
Right.
So they're both natural products, and I think this might be in the abstract or Sabode's abstract. They're all flavonoids, so they're all in your diet. So actually, to Mark's point about diet, you'd have to take a lot in your diet, but you could boost the levels of these and maybe see an effect. But yeah, they're all natural products. Dark chocolate, red wine, grapes. I can't remember what else, Sabode. There's lots of other things. Yeah. What else are they in, Sabode?
All the berries like spinach. I don't remember all the berries, all the fruits like any berries,
apples, mangoes. It will represent in dark chocolate and red wines.
Yeah, you'd have to eat a lot of it though.
Yeah.
Red wine.
So any natural product you eating like that God gave us, you will find that molecule over there.
Yeah. All right. Look at that.
Great.
101.
Question.
Yeah.
I was getting hungry after that. Binds to the RNA. Do we give it a post side?
We do, so that there is a reduction in foci. We should have looked in the HSALR mouse as well, because we see that's where we see the most significant reduction of the RNA. But we do see a reduction in the cell lines. Yeah. Yeah, and we don't actually, so one thing we don't see is when we treat control cells, we don't see changes in splicing. So some of our other compounds like the MPCs, we're actually shifting splicing up because we're shifting MBNL levels up. So for these natural products, we're not seeing any effect. Well, no, have we done RNA-seq yet? We haven't done RNA-seq yet. I mean, I think if you do RNA-seq with almost any compound, you can see things change, but we don't see DM-specific changes. Yeah.
Over here. Hello.
Yeah.
My name is Jay Lee. I'm first-time attendees,
Yeah.
very new to DM. One of those new individuals coming in, like Dr. Thornton said just an hour ago. Thank you very much. I'm very much enjoying it. So, question: did I hear you correctly that this screen is in vivo?
No. Well,
Okay.
it's a cell screen.
Cell
So
screen.
in vitro cell lines, and we screen first in this modified cell line where we introduced the repeats, integrated the repeats from the DMPK, and then we're looking for things in that cell line. And then the hits that look good, we put in patient cells,
Right.
and things that look good in patient cells, we go into the mouse model.
So, given that it's an in vitro screen, even though it may be a little involved, might you consider screening a bigger library
So we-
besides the natural product?
So we have additional natural product libraries. That was the first one that we got our hands on. So we have additional screens ongoing. Yes,
Yeah, you might find much favorable properties.
possibly. Yeah. Okay.
Hey, thanks a lot.
Yeah.